With Dr. Chris Cutler's team. So Chris, Janelle, and Samantha Van Wart from Dalhousie. Chris is a professor in the Department of Plant and Food and Environmental Sciences. He's been a faculty member since 2007, and his work focuses on pollinator risk assessment, insect toxicology, integrated pest management, and agroecology, with specific contributions to the blueberry industry. Today, Chris is going to be sharing a research update on blueberry fruit fly and the spotted wing Drosophila. Ahead, back, pointer.
Thank you. Come on up, girls. I'm actually not going to use this at all. So I was going to speak, but we decided that my two excellent grad students would do all the talking. So I'm just going to do the introductions. Yeah, my name is Chris, and it's great to be here. And we're fortunate to have partnership and support from WBPANS and MyTex for a couple different projects. One is with spotted wing Drosophila and Samantha Van Wart, who joined my group as a summer student, really jumped into the deep end and quickly transitioned to a master's program. She's going to talk about some of the work that she's been doing with that, and she's in addition to the field work that she's going to talk about today, she's got spotted wing Drosophila being reared in our lab. So we got some planned experiments in the lab with insecticides and behavior and heat shock with flies in the lab. So if anybody wants some spotted wing Drosophila, we can set you up. And then Janelle MacKeil is going to talk about the work that she's doing on blueberry fruit fly. Janelle's a PhD student and has been working on that for a few years now. And we're going to give you an update from 2025. So I'll hand it over to Sam first.
Hi guys, I'm Sam. Thank you so much for coming out here today to hear our presentation. I'm looking forward to talking about my work on Drosophila suzukii. Oh, that. This year's work was mainly focused on improving some of the current management practices that I'm sure you are already familiar with, and building upon and enhancing our understanding of Drosophila suzukii right now. So, if any of you guys aren't familiar with or need a refresher, Drosophila suzukii is also known as spotted wing Drosophila, or SWD. It's an invasive exotic fruit fly pest from the tropics of Southeast and East Asia. It's actually pretty similar to a lot of the fruit flies you might find at home on your bananas, so at first glance it can seem pretty harmless. It was first detected in North America in 2008 on the West Coast in California. But it didn't really become prominent in Nova Scotia till 2011. However, we do consider that a pretty fast transmission, given that it was only about 2 years for it to go from coast to coast. We think this is mainly due to the transport of fresh fruits across the continent because the insect does infest the fruit. Because it's a relatively new pest, we're still learning a lot about the biology of it. It doesn't really have any sort of established natural enemies yet, but we have still over 10 years of research at the moment, and results are very promising. So we're improving all the time. A little bit about the biology of spotted wing Drosophila. I have photoed here some of the insects that we caught this summer. The top picture is a male SWD. It's very identifiable with these iconic wing spots leading to its name, while the female SWD can usually only be properly identified under a microscope. If you have access to a microscope, I really recommend you look for that serrated ovipositor pictured there on the bottom right. And the females use that to cut into the flesh of ripe and ripening fruit to deposit their eggs inside, obviously leading to infested and unmarketable berries. The flies usually measure about 3 to 5 millimeters in length. They have yellowish brown bodies and red eyes. And pictured on the bottom left, I have the larvae. The larvae hatch inside of the fruit after about 1 to 3 days, and they spend a week inside the fruit feeding on the flesh before pupating, and then the adults will emerge from the pupae, and the cycle starts over again. And there can be multiple generations of SWD in one season. For this year, for the 2025 season, we did look at 2 different projects. My first project was comparing various types of homemade and commercial traps, and that will be my main talking point today. Janelle and I also did intend to and did perform some insecticide trials in attempts to determine a reduced risk pesticide or product for you guys. But unfortunately, perhaps due to the heat, we're not quite sure. Our results for SWD on that end are inconclusive, but we do have some pretty good results for blueberry maggot, so we'll get talking about that a little bit later. For my monitoring season this year, we looked at 7 sites total. We had 5 in economy and 2 in Caledonia, and we selected these based off of past presence of SWD. Our 5 here in economy are pictured on that map, and they are managed by the wonderful Ron Welton. His fields are about 9 kilometers apart, spanning across near Economy Point, and they're called River Philip Weedy, River Philip, Campfield, Point Field, and Brown Road. You might notice that River Philip Weedy and River Philip are kind of right next to each other. We did take on River Philip Weedy as an additional site because of its abundance of weeds, given its name. We do have a kind of a hypothesis that weeds may exacerbate SWD populations because SWD might use it as a type of shelter. This year, we didn't really come up with any sort of definitive results, but it's certainly not something we're going to rule out. And I have pictured here our sites in Caledonia. We have 2 sites called Dexter and Code, and they're managed by lovely Peter Van Dyke. Peter Van Dyke. They're about 7.5 kilometers apart. And because of the distance from here to Caledonia and how much sampling we wanted to be doing, we were super fortunate to have the hands of Catherine Benedict, Rachel Ricks, and their lab teams to help us with sampling there. For our monitoring traps, we had 3 types of traps we were comparing. The first one, I'm sure a lot of you are familiar with. They're the solo cups filled with 100% apple cider vinegar with the holes punctured around. The second one was a commercial lure called Century, and we hung that inside of a 1-liter cup with a dilution of 25% apple cider vinegar. And the last one is called Trace. It's another commercial lure, and it kind of comes in this little canopy set with a double-sided sticky panel. And all of our lures and traps were attached to stakes and placed in the field. Just to give you a further understanding of our setup, I have a couple of diagrams I drew up here on MS Paint. The first 2 are the pictures of the liquid traps. As you can see, for the liquid traps, we had about 100 milliliters of drowning solution, and we had holes perforated all around the cups for the insects to enter. For the Century lures, we hung them from the top of the lid of the cup, and we diluted the apple cider vinegar to about 25%, just to make sure that the main attractant was the Century lure and not the apple cider vinegar. And then here's a kind of an understanding of where we put the lure on the canopy for the panels. It's kind of stuck underneath there, and then it's hung from the stake. And the panel is then double-sided because of the canopy, and that's kind of the theory behind it, as well as it's meant to protect it from wind and rain. And if you guys are interested in purchasing the Century or Trace lures, you can get them from either Great Lakes IPM or Solita. A quick map of our trap layout. Our traps were all about 20 meters apart and 3 meters from the forest edge near the woods. And they were kind of ordered randomly just along the edge of the field. We sampled about twice a week, where we replaced our apple cider vinegar regularly each collection, as well as our damaged traps and lures when they needed to be replaced. When the berries were ripe enough, we were able to collect about 3 cups of berries per site each time in attempts to do some larval extractions. All of our capture was then quantified in the lab under a microscope. And it should be noted that for our panels, because of the difficulties in identifying the ovipositor for the females on the panels themselves, we only quantified the males on those panels. I have a figure here of the total SWD we captured and counted this year. From the end of June to the 23rd of September, we counted over 9,200 flies total. But you may notice in this figure, they don't really start to pick up the numbers until about late July. So we're just going to zoom in on those figures right here. You might also notice that when I broke it down by sex, there appears to be more males than females that we caught. But this is just because we only quantified the males on the panels and not because there was any sort of sex discrepancy. And you might notice that I have the harvest periods blocked out in the middle for Caledonia and economy. And we have some peaks, which will become a little more obvious in some of my later figures. But these peaks in SWD populations don't really become more prominent until after the berries are out of the field, like post-harvest. So it's definitely something we are going to be looking into. For the average SWD caught per trap, again, you can see those spikes in the population a little more obviously. That one in the middle was mainly driven by the Caledonia fields because those fields are more south and therefore ahead in ripening. And then at the end is our sampling from economy, and they just take off around the 4th of September. For each type of trap, the trace traps, the panels, were almost consistently able to catch more SWD on average than the ACV and the Century traps. And this is especially obvious near the end of the season, where you can see that the trace lower panels on average caught almost or more than double the amount on average than the liquid traps. We think this might be really useful for you guys in case you want to do some early season monitoring, since the trace traps are a little more sensitive for capture, because the only thing the flies have to do is smell the lure and get stuck on the trap, versus the liquid traps where they have to actually climb into the holes and then drown. But those might be a little more useful for if you're doing some full season monitoring. And lastly, I have here a figure for our average SWD caught per location. Again, you can see where I have the Caledonia sites pictured here, and that's kind of right after we stopped monitoring there. And then the economy fields a little further on where they start to take off. Even though, remember in our maps that the fields are only a couple kilometers apart, we have a lot of variation in the amount of average SWD caught in each location. So we think it's really important that you continue monitoring in each of your fields and not just select one field to do monitoring in and consider it an average. And then for our larvae extraction process, we used two methods. One was the brown sugar flotation method, and the other was the salt extraction, where essentially we just kind of crush the berries and soak them in different solutions so that the maggots come out. The reason that I'm not really covering a lot of the results for our insecticide trials was that in total, we processed about 160 liters of berries from both the insecticide trials and just the 3 cups of berries I caught for each collection. And after all those berries, we only found a single SWD larvae. I mean, it does kind of align with the fact that we notice that all of the flies don't really come out till post-harvest when the berries are gone, but we definitely expected more than one single larvae. We were very successful in extracting plenty of blueberry maggots, though, so it's definitely not an error in our process, which Janelle will definitely talk about the results for that in her talk. If you guys do want to try some of the extractions on your own time, it's important to know the differences between the SWD and BFF larvae. You can notice here for the picture on the left, the SWD larvae has 2 tapered ends, while the BFF larvae only has a single tapered end and kind of like a rounded or flat end. The SWD larvae usually measures about 2 to 4 millimeters long, and the BFF is normally about, or can be up to 8 millimeters long. It just really depends on the instar for each of them. We were very careful when we did do our processing, so no worries there, but we do acknowledge that it's not incredibly obvious, so always be careful if you are doing any extractions on your own time. We did quantify some of the damaged traps for this year. In total, we counted 31 instances of traps damaged. with the panel traps being damaged or interfered with the most. A lot of this was due to elements like wind and rain, because the panel is just kind of hanging with the canopy. It kind of swung around a lot, especially during unpredictable weather, as well as animal interference being noted for all of our different traps. This is because the lures, whether it's apple cider vinegar or any of the commercial lures, they smell a lot. And they're brightly colored as well, so the animals are naturally curious, and they will go and ultimately end up damaging the traps by accident, which might be something to keep in mind, especially when you want to consider the cost of the traps. A couple pictures here of the damaged traps and other things that have to do with the convenience of managing them. The three pictures in the middle are very clear evidence of animal interference. We have everything from a crust trap to like punctures in cups. That left picture there is just a panel that got detached in like the best case scenario. Sometimes we'd find it flown off in the middle of the woods, and remember, they're sticky and it's very inconvenient to have to pull them off of all the leaves and the grass, and you are going to be losing some insects during that process as well. This doesn't always happen, but it was very frequent, so might be something you want to keep in mind. And then on the right, I have pictured this kind of scum that can appear with the apple cider vinegar traps. When they're left out in the heat for a long time, they can kind of get a little bit murky, and the insects can end up getting caught within this kind of stuffs or scum or whatever. So it might be something you want to consider when doing your analysis of what you actually caught, because it could be a little bit tedious to go through. We did quantify some of the efficacy and convenience stats for the 2 dates pre-harvest, which we know are important monitoring dates for you guys. As you can see, the panels, traps at the bottom, they had the highest capture rate, which is no surprise there, given how easy it is for an insect to get caught there. But they are ultimately the most expensive, about over $25 a trap each, and they have a little bit of cumbersome maintenance. However, I guess the ID is pretty easy to handle since you don't have to go through much. You just need to look for the 2 spots for the males on the traps. So depending on what your priorities are there, something to keep in mind. And then for the standard apple cider vinegar traps, which I'm sure lots of you have used before, they actually did pretty well compared to the commercial traps. They had the highest selectivity, meaning that they could just catch the most SWD versus other random insects. And they are definitely the most cost effective. But again, you are dealing with that possible ID process of sorting through that murky apple cider vinegar. And then lastly, we just have our Sentry lures. We kind of consider that maybe a happy medium. If you're looking for something commercial, but you're not looking for something as expensive as the trace lure and panel, definitely something that would work for you. Our plan for 2026 is definitely going to be repeating the insecticide trials and repeating the monitoring trap assessments. We do want to add some variation to where we placed our traps this year or for next year, whether that be in the woods to the middle of the crop field, because we only did the edges this year, as well as we want to modify perhaps the trace panels to just being against the stake. While we might lose some surface area for the double-sided panel aspect, it will definitely be better for it not swinging around from the weather so much. And lastly, our big question still is, why are there so many SWD when there's no berries in the field? Because all of our numbers were found post-harvest, and we still can't find any larvae in the berries either. We definitely don't doubt that there's plenty of evidence that the SWD go after the berries, they infest the berries, they've been a pest in North America, and they are economically damaging. But we don't know why here in Nova Scotia they're really only coming out till after the berries are gone. So we do want to definitely spend some time looking into the ecology of the SWD and figuring out where they're going for the majority of your season, which will definitely help you better understand timing for your harvests and your monitoring. This is all I have for spotted wing Drosophila, so I'm going to hand the mic off to my colleague Janelle.
Do you have the clicker with you? All right, hello everyone. Just gonna go into this quickly because I don't want to hold you up from lunch too much. But following Sam here, my project has been heavily focused on the blueberry fruit fly, also known as the blueberry maggot. But there are a few different projects that I've been working on over the past few years. But today, the two that I wanted to talk about are some updates on the development of the degree day model for the fruit fly, as well as talk a little bit about some of the different insecticides that I have been testing over the past few years. So to get right into it, we have the larvae here on the left, and then also known as the maggot. And then right beside it, we have the adult blueberry fruit fly. And if there is any one good thing to be said about the fruit fly is that it has that very unique, very distinguishable wing pattern. I say good only because it can help us identify it on our traps, because there's really not going to be anything else like that that you're going to find on your traps. And then on the far right here, you're going to see what you would find if you were to open up a berry and find a maggot on the inside, but obviously, hopefully, that's not the case for you. Oops. And so a site that you're likely a little bit more familiar with here are the yellow sticky traps. And if you check these in the middle of the summer, you likely go out and see them and see a ton of different insects stuck to them. And on these three traps here, there's actually quite a few blueberry fruit flies stuck to them, but even from this distance, it can be tough to tell. So we can zoom in on this middle trap here, and then we start to be able to see that those features kind of flash out at us. So we have a fruit fly here and here and here. There's quite a few of them. I won't point them all out. But also what they have is this little white spot on their back. So those are the two main things that you can look for on your traps, is that W shape on their wings as well as that little white spot on their back there to help you out. And fruit flies are a little bit different from spotted wing, and in one of the ways is that they only have one generation per year. What this means is that when they come up out of the ground, they emerge, they're active for about a month, and then they die off. They don't come back again until the next year. And so if you know that you have fruit flies in your field, you might wonder, well, where are they at right now? Right now, we are at, again, I'm sorry, I'm favoring this side. I don't want to hit you. But this bottom portion of the cycle here. So these pupae are in the top inch of your soil, and they'll stay here for the remainder of winter, not developing, not moving at all. But then as it begins to warm up in the spring, we see those pupae slowly start to develop. And then at the end of June, beginning of July, we're going to see them emerge as adults. And once they emerge, they'll take about a week or so to mature and mate. But then once they are mated, they will immediately be ready to start depositing their eggs into any ripe fruit that they can find in your fields. And these females, they can deposit anywhere from 25 to 100 eggs in their short lifespan. And what they'll actually do is they'll leave behind a pheromone or a scent over top of the berry that tells all other fruit flies there, I've already put an egg inside this berry, don't put yours in here, go after the other ones. And so left unchecked, these populations can get out of control pretty quickly. But now you have an egg in your berry. And so within a few days, that egg hatches and it results in that little maggot. So that maggot will then begin moving around inside your berry, consuming the pulp, growing bigger. And so then come harvest time, either that berry gets harvested and it goes off to the shed with the rest of your berries with the maggot inside, or the larvae may drop out of the berry down to the soil where it digs its way into that top layer and pupates. And so that cycle will repeat itself year after year, and that's pretty consistent. But what's not always consistent is the timing that each of these stages are happening. So the time of year that those flies are emerging and starting to deposit eggs into your berries. And the reason for this shift, it has a lot to do with temperature. And this is because insect growth and development depends upon temperature. And so we have a lower developmental threshold for these insects and an upper developmental threshold. And for the fruit fly specifically, that lower developmental threshold is 4.7 degrees Celsius. So below this temperature, that pupae in the soil won't develop at all. But they also have an upper developmental threshold, so when it gets a lot warmer, they're going to either slow their development or completely stop. So for these flies, we really do have a sweet spot for development. And so as that temperature starts to increase above 4.7 degrees Celsius, we see their rate of development increase until they are finally able to accumulate enough heat units or degree day units to finally be able to emerge. And so as you know, we get variable weather from year to year. And so let's say we have a warmer spring on average. We might see those fruit flies emerge a little bit earlier because they've already accumulated enough of those heat units. Or if we have a cold and wet spring, we might see them emerge a little bit later. And so that's why degree day modeling is so helpful, because it's going to help us calculate that to be able to predict different points in the insect's life cycle, such as when they're going to emerge. But it can also help us increase our efficiency and our precision of scouting efforts, such as when is the best time to put our traps out in the field and when should we be checking them. But on top of that, also optimizing our management decisions, such as when is the best time to be applying our insecticides. So there have been fruit fly models developed in Maine as well in New Jersey in the early 2000s, but it really is important for us to be able to determine whether those models represent our local population for our climate here in Nova Scotia. And so what I'm doing is I'm using the province's suite of in-field weather stations. And if you have one of these, you're likely familiar with all the things that they can do. But they're going to collect data such as air temperature, soil temperature, moisture, humidity, precipitation, wind speed, leaf wetness, etc. I won't list them all, and they do this every 15 minutes of every day. So they're very, very helpful for the development of our degree day models. And so in some of these fields, we place these yellow sticky traps along the field edge. And then from June until August, we go out and we check these 3 times weekly for the number of flies that we've found on our traps. And so here is showing some of the data that we've collected off of these traps over the past few years. And you can see that the pattern is fairly consistent. They're going to find them emerge the end of June. They're going to peak mid-July, and they're going to taper off by the end of July, beginning of August. But at the same time, you can also see that those populations do shift around a little bit from year to year. And so they can drift by a week or more depending on the temperatures that they're experiencing in the fields. And so here are some of the fruit fly emergence thresholds that we've calculated over 2023 and 2024. And I'm working on the 2025 data, so it's not here yet. So I will say this model isn't complete, but it still does paint a very good picture for us. And so we see that the first emerging flies happened around 570 growing degree days, and the population was essentially finished emerging around 900 degree days. And then when we take a look at the range for first emerging flies, you might think that that's a little bit big, but in the summer, we're averaging around 15 to 20 degree days per day. So it's a range of about a week or so. But what this is really telling us is that in our fields, the earliest emerging flies are going to be around that 500 degree day mark. So that would indicate to you that I should get my traps out now and begin monitoring them, because this is the earliest that I may find them. And this range is extended a little bit, and that's because sometimes it takes a little bit of time for those fruit flies to get from sprout fields to crop fields. So those first captures that you're getting, they might be a little bit delayed. And as I mentioned, there were models that were developed in other areas. And this study looked at the number of degree days to median emergence for the fruit flies. And they found that in New Jersey, it took 934, and in Maine, 971. And so what's important about having those ranges that I showed on the last slide and this information is that when we look at our data, we found that it was a little bit less. The number of degree days to median emergence happened around 739 degree days. And so this doesn't mean that there's anything inherently wrong with the model, but it just means that our insects have adapted to our environment, our local conditions, and align closely with our crop. But if we had relied on that main model, by the time that our fields were hitting around that 970 degree day mark, our fruit fly populations had already completely come and gone. So we really would have missed our optimal management windows. So it's just driving home the point that it is important to have local degree day models. It's in Nova Scotia. But again, just so I can show you, is that this happens in other insects. So this is with the apple maggot, which is very similar to the fruit fly. This study took a look at their first emergence, or how many degree days it took to first emergence in the same year in different locations. And so in Utah, they found it took 809 degree days. But then in New York and Ontario, it was a little bit less, so 628 and 638. So depending on the location that you're in, the insects will adapt differently. Again, showing us that we need these Nova Scotia specific degree day models. And so before we get the model up and running, I wanted to show you a way that we could potentially use this information. And so Hugh and Dr. White have been working on degree day models with blueberries for the past while. And what we can do is combine some of that information. So we can see that this is using the 2024 data as an example, but our first emerging flies in 2024 happened around June 24th and 500 degree days. But then we can cross-reference that with the blueberry data and see, well, I don't have any blue fruit in my field yet. And so that notes to you, okay, flies are starting to come out, but they're not a risk to me yet. And I don't need to apply insecticides likely quite yet, because I know that those fruit flies can't deposit eggs into unripe fruit. It's just too tough for them to get into. But we can also see that risk period increase considerably when we see that 50% of the population has now emerged, and you now have 10 to 30% of ripe fruit in your fields. And you know that these flies are very good at finding that ripe fruit. And what I wanted to note about this 99% emergence, and this was around July 24th this year, but through the past few years of monitoring these flies, I see the same thing time and time again. It's that middle of July is when these populations are peaking. And by the time that July 24th comes around most years, I'm getting significant declines in my counts on my traps. So all I want to say here is that if you are waiting a bit longer to put your insecticides on, and you know that you have fruit flies in your field, it's likely that you may have some infestations if you aren't applying a little bit earlier, that you might have missed that optimal window. And I know that I just shot a lot of different degree day numbers up there, so I want to show you what this might look like in the future. And so Perennia has on their web platform a degree day calculator, which you likely know about. So you can come to it and insert a station. This can be your station or whatever the closest station is to you. And you can put in a start date. Here I have January 1st, and an end date I just put around harvest. And then you put in the base temperature of 4.7 degrees Celsius. So this is a base temperature again for the fruit fly. We can then click calculate. And what this is going to do, it's going to generate a line for us showing our cumulative number of degree days over time. And we can take our cursor, we can hover over this line, and we can see the date and the corresponding number of degree days that have accumulated in that specific field. So what we're looking for in our fields is when we start to approach that 500 degree day mark, that's when we want to get our traps out in our fields and begin checking them. But what we can also do is come down here to the fruit ripening data. And like I said on the previous slide, we could put this data side by side. So we could see, okay, my fruit fly population is starting to come out, but I likely don't have any blue fruit in my fields yet. Or okay, now 70% of my fruit fly population has emerged, and I have 50% of ripe fruit in my fields. And so it might not look something exactly like this, but in the end, when we finish the model and get it into your hands, we hope to make sure that it's very intuitive for you to use. And there's just two more things that I wanted to note on this before I touch on the insecticide stuff. And the first being is the movement of blueberry fruit flies across split fields, or fields that are nearby each other but in different production cycles. So we can see earlier in the year, we see that the fruit fly populations start out in the sprout field. And this is because they were in crop, they went down to the soil, and when they come up again next year, they're going to find themselves in that sprout field. So that's where we're normally getting our first captures. But then they're going to realize there's no crop here. There's no fruit for me. So they're going to go off searching for fruit. And if you have split fields, they're going to easily find that fruit. And notice that that shift where they're going from sprout to crop happens exact time that your fruit is starting to ripen. So they notice this and they can smell this. And so they move over to those areas. And here is, I know it's not something that's easily changed or fixed at all, but I just wanted to highlight that these fruit flies really thrive in a system where they have access to berries year after year, compared to if you had your fields in the same production system, they would be essentially starved of those berries every second year, making it difficult for them to thrive or even survive there. And this degree day model was made using soil temperature. So I know that a lot of you, or some of you, might have your weather stations, but not all of them have the soil probes. And so what I went ahead and did is I graphed the cumulative number of degree days using both the air temperature sensor and the soil temperature probes. And what I found is that early in the season, we see that air temperature lags behind a little bit, but by the time we were seeing first emerging flies, it was almost identical. And so I'm showing this to show that if you don't have a soil temperature probe, but an air temperature sensor, it's likely that this model is still usable in your fields, but just keeping in mind that the model was made with soil temperature data, so the soil temperature probe is going to be the most reliable method. And getting into some of my insecticide studies, so I've done some small plot field trials over the past few years, and this is an example of some of my 2024 data, but it really does show some of the similar trends that I've been seeing over the past few years. And that's that we consistently see the lowest number of maggot counts when we're applying Assail and Savanto Prime. But kind of like Dr. White mentioned, we do get a lot of variance in our data, and we don't actually have a lot of significant difference between a lot of these products here. And that's a good thing, because it means that there are quite a few products here that we can use that will work against the fruit fly. And in 2025, I ramped things up a little bit. I tested some more products and a few different combinations. And I won't get into each one of these products here, but again, showing that Assail and Savanto Prime are working very well, along with Cormorant and Exferil. And if you apply insecticides for fruit fly, you're likely familiar with at least one of those. I know a lot of people use Assail right now. And so what I was interested in this year was in fields that have high infestations, does doing a second application make a difference? Is it going to help with this population? And so I tested that with Assail and Savanto Prime. So we did one application, so that's your 1x, and two applications is your 2x. And for Assail, these applications were spaced out by 12 days as per the label, and for Savanto Prime, that was around 7 days. And what we found is that it didn't make a significant difference, showing us that one well-timed application is good enough. And so that was really promising data to see. And this was another trial I had this year where I tested a lot of the same products. When we see the same thing, we see Assail and Savanto Prime worked very well. Exferil and Cormorant are still here. And that one application versus two applications, again, didn't make a significant difference. And I wanted to talk about how out of control some of these infestations can get sometimes. And so these were some berries that I harvested around August 1st from an untreated area of a crop field. And so what I do is I go out to the field, I harvest the berries, I bring them back, and I measure out 1 liter of berries and put them into this container here. I then will gently mash the berries just enough to be able to break the skin, and then I'll pour a brown sugar solution over top. I know this sounds like an elementary school experiment, but it definitely does work. This causes the maggots to then float to the surface where I can collect them and count them. I found 96 maggots in just 1 liter of berries. And so this is obviously an extreme example, but at the same time, it can happen. And given that I harvested these berries around August 1st and the size of these maggots, it's likely that these eggs would have been deposited in mid-July, again taking us back to that degree day modeling and how helpful it may be. And I wanted to end on this slide here. So here are a few different products we've been testing over the past few years. This is definitely not a full list. We've been working with bioinsecticides, some new chemistries in the pipeline, some proprietary products, but what this table is doing is it's keying on some of the differences that we're looking at. So we have these different products from different insecticide classes with different relative costs, but they also have varying effects on the environment or non-target organisms as well as humans, and of course how efficacious they actually are against our target pest. But we also see differences within the same class. For example, Assail and Savanto Prime, they both work very well against the fruit fly, and they're both group 4 insecticides and have similar relative costs. But where they differ is their non-target toxicity on non-target organisms. And so I did a study where I went out to the field and I collected field crickets from blueberry fields and brought them back to the lab. And if you don't know much about field crickets, they are almost guaranteed in your blueberry fields, and they're known to be avid consumers of hair fescue seeds. So obviously a great thing to have in your fields. And what I did is I tested the field rate applications of these insecticides against the crickets. And I found that a field rate application of Assail killed these crickets almost within an hour, and a field rate application of Savanto Prime, these crickets just continued to live life on as if nothing had ever happened to them. And so you can see that reflected in the lower rating, ecotoxicity rating for Savanto Prime compared to Assail. But it's not just the field crickets that are important. You know, there's things such as ground beetles and wild bees and queen bees and worker bees that are still out in your fields when you're applying these products. And so the main thing here is first and foremost that we're going to find a strong and reliable product that's going to work against the fruit fly. That's the most important thing. But keeping in mind that there are other things to consider when looking at keeping our fields sustainable over the long term. And I wanted to end there, and I'm happy to answer any questions, but first I just wanted to give a quick thanks to WBPANS and MyTax for their support in this research, as well as the WBPANS Graduate Research Awards provided to both Sam and I, the agrochemical companies for providing products for us to test in our trials, our summer students this year, Leah and Paisley, as well as Catherine Benedict, Rachel Ricks, and their teams. They helped a lot with Sam and her spotted wing work, so we greatly appreciated that. And all of the growers who allow us to use your fields for our trials. We greatly appreciate that and always love stopping to chat in their fields. But thank you very much.
I'll take time for 1 or 2 questions if there's any questions in the room.
So on the blueberry maggot, was that full field application or perimeter spray?
Yes, so that was not a full field application. What we do is we do these small plot trials in production fields. And so our plots are quite small, but then we do replications of that in multiple fields. But it was not full field application or a perimeter spray, just small block trials, kind of similar to the work that Dr. White would do with the herbicides.
And on the SWD, was there any reason or consideration to using the yeast formula versus the apple cider vinegar?
I'm going to leave that one for Sam.
We definitely think the yeast could still work. We didn't really think of a difference between the yeast or apple cider vinegar. They're just usually looking for something fermented that smells. So yeast could easily be an option for you if that works better for you.
You probably have less damage with animals and stuff with the yeast as well. Thank you.